Existing fluorescence recovery after photobleaching, FRAP protocols, are either incomplete or overly complex. The protocol presented in this video is significant in that it is comprehensive, practical, and straightforward. The main advantage of this FRAP technique is that it allows for the quantification of protein mobility within and between subcellular compartments in live cells.
FRAP is being studied for both therapeutic and diagnostic purposes. One such use is to quantify diffusion within drug delivery systems in different tissues in vivo. This information can be used to optimize a drug structure and composition.
The FRAP microscopy technique has provided insights in a broad range of fields in which understanding diffusion phenomenon is of interest. These include cell and molecular biology, developmental biology, neuroscience, material science, advanced biomaterials, and drug discovery. Although the protocol described is for use with mammalian cells, the concepts can be applied to other systems, such as plant cells, yeast cells, and bacteria. An individual who has never performed this technique may struggle in determining the optimal image acquisition parameters, creating an acquisition region of interest that is suitable for their experiment, and calculating the mobile fraction and half-time of recovery. When attempting this procedure for the first time, be sure to perform several pilot experiments to optimize the protocol in the context of the aims of your experiment.